But now we will move into the last set of presentations for today. So there'll be two back to back that will cover receptor tools. So first we'll hear from Dr. Raphael Trevizani on the TCRMatch tool, and then we'll hear from Dr. Mahita Jarjapu on the BCRMatch tool. So together they're kind of two sides of the same coin for receptor tools, so to speak. So we are running a couple of minutes ahead of schedule, but no harm in starting early. Raphael, welcome and take it away with TCRMatch.
Nina, can you just confirm to me that you can see my screen and I removed all the artifacts?
Yes, it all looks good.
Great, wonderful. So hi, everyone. So I'll begin the presentation on TCRMatch. And before I tell you about TCRMatch, the tool, let's begin with a little bit of biological background on T-cell receptors. Now, you've seen these figures here during the workshop in the presentations about peptide binding today earlier when they talked about MHC1 and MHC2. Now, in this presentation, I want to focus on in this part here, the part where the T-cell receptor recognizes the presented peptide as an epitope. Now, let's use a different figure that details the TCR a bit more. And there, this is the T-cell receptor. It is a heterodimer where the alpha chain is encoded by a series of genes that undergo somatic recombination prior to producing the TCR. So here is the segment called the variable segment, marked with represented by the letter V. And here you have the J segment, the joint segment, and the constant segment. And the second subunit of the TCR is the beta chain, which is encoded by certain gene segments, just like the alpha chain. In the case of the beta subunit, you have the variable, the joint, and the constant segments, just like in the alpha chain, but you also have another segment called the diversity segment. Now, both alpha and beta chain contain three loops called the complementary determining regions that are encoded here. In the case of the beta chain, they are encoded by the VDJ gene segments. So you have three loops that are called the CDRs 1, 2, and 3 of the beta chain. And all these loops are encoded in this VDJ region. The CDR1 and 2 are encoded in the V region. And the CDR3, as you can see here, it's the only segment that spans from the V segment through the D segment all the way to the J segment. And it's generally considered to be the most important because, as you can see, it's the most diverse. And it's also the one that most directly interacts with the presented peptide. So because the CDR3 beta directly interacts with the peptide, it became focus of most research. And one technique that has become very common in T-cell biology is called repertoire sequencing, which is the sequencing of the TCRs of a population of T cells. And these are actual sequences of CDR3 betas. And analyzing these sequences is very interesting because it's a lot of diversity for a relatively small region. And it is very small. Like you can see here, there are 10 mers or 11 mers. And they're usually flanked by a cysteine and a phenylalanine, sometimes a phenylalanine in the end here. You see a tryptophan in this phenylalanine, but it's usually very conserved. Now, let's imagine that you have the sequences of the CDR3 betas of a population of T cells. That in itself tells us nothing about the epitopes that these TCRs will bind to. So how do you find out which epitopes these TCRs bind to? Well, there's an online database that contains curated T-cell epitopes for us to use. The IEDB website contains a series of sequences of CDR3 betas and their associated epitope sequences. So we can search the IEDB for the TCR that matches the ones that we have, and we can see their corresponding epitopes. So it's a matter of taking each sequence of the CDR here and search the IEDB for the sequence. And once you find the sequence there, you know the corresponding epitope. Now, that's very easy because in the case of this sequence, we were lucky. We had the exact match in the IEDB. But what about this sequence? What if the sequence is not in the epitope, in the IEDB database. I would still like to know which epitope it binds to. So what we can do is use the sequences that we have in the IEDB, for which we know the CDR3 beta, and compare it to our query. And if the rationale is, if these sequences are similar enough, then they should bind to the same epitope. Now, then the question becomes, how do we compare sequences? How do we match? How do we compare if the sequences are similar enough? So our team tested many algorithms, and I'm going to show you one graph from the TCRMatch paper that I think summarizes the result. It's a graph of the precision recall curve. So the way you read this graph is the best result to the right of the graph, and upwards is also better. So when they tested all these algorithms for the one that would best capture the right CDR3 beta and the epitope pairs, this is the result that they got. And this one here that I'm highlighting is the one that became known as TCRMatch, is the one with the best result. Now, how does it work? The table here, this table is from the paper. And if you read the last row, it says, it performs a comprehensive comparison of all possible k-mers using a Blossom 62 matrix. So what does that mean? It means it's a k-mer approach. And if you don't know what a k-mer approach is, or you need a refresh, let me explain it to you. First of all, TCRMatch uses the Blossom 62 matrix, this one here. So given two CDR3 betas, the one at the top and the one at the bottom, how similar are these CDRs according to the k-mer method? So we're going to start with k equals 1, and we'll gradually increase the value. So k equals 1 means we're going to take each amino acid separately, and we're going to start from left to right. And so the entry is AA. So in our Blossom 62, A and A means +4. So now we take the bottom sequence and we shift one amino acid to the right. So that gives us A and S, which in the Blossom means +1. And we shift right again, is AS again, is +1. And we shift again, and AI means -1. And on we go all the way to the last amino acid in the, at the bottom sequence. So AY means -2. Now we go to the second amino acid at the top sequence. So we have S and A, which is +1, the whole thing all over again, using the second amino acid from the top sequence, all the way to the end of the second sequence. And once we reach the last amino acid of both sequences, we're done with k equals 1. So now what we do is switch from k equals 1 to k equals 2. And now we're going to compare each of the dimers. We'll start with AS matching AS. So that's A and A +4, S and S +4. Shift the dimer at the bottom and to the right, and A and S +1, S and S +4. We shift right by 1 again, A and S, S and I, all the way, shift again and again, and we keep comparing all the way to the last dimer. You got it right, all the way through. And then we shift the top sequence to the right, and we restart the process. And we're done with k equals 2 once we get to the last dimers. And then we switch from k equals 2 to k equals 3, 4, 5, all the way to k equals 10, in which case we only have these two peptides here when k equals 10. And in this case, we're using k equals 10 as the maximum k-mer because it's the size of the smallest sequence. So we can't go above that. So the final score is the sum of all these entries that kept popping up here as I was running the k-mer comparison. But we normalize the score. So you're going to be presented when you use TCRMatch with a normalized value scaled between 0 and 1, because what we do is we divide the score or the sum of the k-mers by the self-matches of these two sequences. So you're always guaranteed to get a value between 0 and 1, 1 being the perfect match. So let me just demonstrate very quickly how you use TCRMatch using our website. There's just a basic function. What you do is you paste your CDR3 beta sequences here, or you can also, if you have too many, you can also upload them from a file, and you can select between using the IEDB database or a cancer-specific database from CDAR, and you can set different thresholds for sequence similarity. Maybe we recommend a 0.97. Maybe you want to use something more lenient, or maybe you want to be a little more stringent. So we recommend a very stringent value of 0.97. And if you run it, this is the result that you'll get. You'll see this table here. And here in the first column, you have your CDR, the CDR3 betas you submitted. Here you have the matching CDRs found in the IDB. And here's the measure of similarity between them. So this is the score that varies between 0 and 1 that I told you about. And this is the set of epitopes that your sequences, these sequences here bind to. So probably similar sequences will also bind to them. And there's also extra information here about where we got these epitopes, and so the source organisms, like where these epitopes come from. But that's it. That's the standard output that you get when you run TCRMatch. Now, TCRMatch, if you're interested in more details, I recommend you to read the paper. It was the brainchild of many smart people, and it's currently in active development. We're trying to make it better, improve some features, and hopefully in the future I'll be able to present them. So I'll pass it back to Nina.
Awesome. Thank you so much, Raphael, for that walkthrough. Much appreciated. And then we'll move straight into the second receptor tool for today. So this is BCRMatch by Dr. Mahita Jarjapu. Mahita has very kindly let me and wants me to acknowledge that she is deaf. So please do follow along with her presentation. But if you have any challenges following along, her slides have all of the information there. So this may be a good time to look at your screen if you're maybe multitasking or whatever you might be doing. So yes, please do follow along. And with that, Mahita, I would like to introduce you to the virtual stage.
Thank you very much, Nina. So you can see my screen.
Yes, looks good. Perfect.
Hi, good afternoon. So my name is Mahita, and I'm a bird dog in the Peter Lamb, and I work with the IEDB. And today I'm going to be talking about PCR, which is a tool to predict antibodies that bind to a common epitope. That's all. A PCR, I don't know, it's a command line too. At the website you go to command soon. And this tool is a pre-trained machine model to predict a period antibodies say an epitope or not. And so here you have antigen, you have a epitope, and we want to know the two antibodies A and B, and if you want to know whether they bind to the same epitope, that is when you can use PCR. And I don't know the two the sequences of the serial episodes antibodies. And the model that I mentioned here, they are actually trained on antibody pairs that have been experimentally validated to say an epitope. Now, as I mentioned earlier, as you have heard to say, talk about the next generation tools, this is what we have in mind, that the web server implementation of our tool. And we hope that it will be up over the next 3 months. Now I'm going to take you through how the web server should work. So here you have a part where the user will provide the sequences the serial episode antibiotics, that without episode. The year and the names of the antibiotics are 1, 2, 3, 4, and 5. And then and the sequences of each of the serial that the light turn and the heavy turn. And then he had turned to select time to learn model. And he had also year 3, but in that we have provided time to learn models. And then you can tell and we have also provided the train data. And then this is how the output will look like. And what happened to the server uses a pre-trained model to predict what antibiotics are the same epitope with the antibiotic of interest. And then the either or is prediction or in a learning model is listed in a table. As you can see here, the column antibiotic per indicates the per of antibiotic and then the prediction. So, at to point out that to be updated. It's a common at the Cairo for order version. So somewhat that we then be modified when server is actually implemented. Now come back to the command line version. The better start what you need is that you need to ensure the Docker is installed on your computer at the Docker engine learning. And then there are 3 methods installed with the command line too. That's one the pre-Docker image. And then the second is by the Docker image. And the third one the local installation. And I recommend use the pre-Docker image. And I'm going to talk about the first method of installation. So the first step, what you do is pre-build the pre-Docker image use the following command line. So this is the example, so this is the command that I use to build the Docker image sent to the server. And then basically a pretty latest version of BCRMatch center server. And then the following line indicates that system was successful, which is the line. So when the execute command, these are the lines that will appear. And here you can see the status, say that it is up to date for this web server. And then the second step is you can tag the previous Docker image with the name used the command line. So here, what in the command, I'm tagging the image, the BCRMatch image with the name BCRMatch. Basically, it is about giving a name to the local version of the image that you got from the server. And then the second step, how do you access the container? So you can access the container by the command Docker and I do BCRMatch pin path. And then the second internet step, you have to make sure that the container is present and you can check the content of the container by using the list command. So when the access the container, it takes you to inside the container. So that is why you see a different here. And within the container, you see the following file. With the different files that are needed to run the program and to the page folder. We contain the input file. And I'm going to explain more about that. Now, the next step is to take it input file the present the page folder. And the use the command cd example, then it will take you inside the example folder. And then you can use the ls command to use the content of the folder. And we can see that the input files are present. And now I'm going to explain what are the different input file formats. The input file format can be either in a TXT format between upper and body. That is how the TXT format will look like. And the third column at the third column is the name of the antibody, equal to the ID, to the one to find. And then the each antibody and the sequence of each of the CDR loop. That is the sequence of the CDR1 loop and to the sequence of the CDR2. Likewise, the sequences of the CDR3 loop. That is the each of the time antibodies. And then the input can also be displayed factor files without the bird. The UN factor file called CDR sequence dot factor. And here the antibody name or antibody ID. And the sequence of the CDR name. And this is antibody 2. And the sequence of the CDR loop. And here this is the CDR2 sequences. And you can see that the antibody ID should be the same. And here you have the sequence of the CDR2 loop. And here you have the CDR3, the sequence of the CDR3 loop that the antibody do. That is how the type will look like. And this is the return CDR sequences. And likewise, our similar structured files that allow to turn CDR sequences. Now, the next step is I want to show you how we can run BCRMatch using the TXT file format as the input. And we use the command Python and run this code. Run BCRMatch.py. And these are the third argument. And I'm going to explain what do they mean. So I specify the input file, and this is the path to the input file, and this is the name of the input file. And then TN specify the train data set on which you are the learning model to be trained. And here give it the name and let it embed as the name of the train data set. And then specify that the output should be same into your file. If you do not specify that output, then if you not specify the argument, then the output will be printed to your screen. And this is the name of the output file. Now let us see this is what happens when you run the runtime execute the command. This is what appears on my screen. It starts the program and then it shows the progress and then finally it ends with the completed word. And then now how can we see the output? So I'm going to say how we can see the output within the command line. you can use the V command to V at the name of the output file. And the output file contains the prediction code from each of the file classifiers and percentile for each prediction code. And this is how the output file appears when I open it on the command line. So what do these mean? So you can see the column name. It's open Microsoft Excel. You can read like a table, which is separated by commas. Now to the third column. The third column is the antibiotic pill. And here, one is the name of the antibiotic pill because the compare between antibodies. One and one basically compare the same antibody. And then the second column is the prediction from the random theory classifier. Here you can see the prediction column. And then the third column, which is percentile rank. It indicates the percentile rank or the prediction from the random theory classifier. And similarly, the third column is a prediction made by the logistic equation modeling model. And the next column is a prediction, it's a percentile rank of the prediction from the logistic equation classifier. The last other column that contains the prediction from the other models like the CNP, CV, and other advanced models. And then I'm going to show you how you can run PCRMatch using the bird file files as input. In the previous command, we used the DHC file, but here we are using the target files as input. So in order to do that, you need to use the command -cs. So basically, you run the same command Python, run PCRMatch. But the input is different. Here you can see it at the specify the name of each of the target files contains the return sequences. And then minus here is again the target sequences of the serial and the sequences of the light turn serials. And here I'm giving the output. I don't specify the name of the output on CAD. At the train data set, again, build listed. And at the happens when I want to monitor the progress. The ones I run the command, that is what appears on my screen. And then I can list the contents of the screen. And now I can see that the output file that I specified here is now present in the folder. And then when I open it, you can see the same output that I explained earlier. So with that, this is how the command line works done now. And there will be to get the web server up very soon. And this is a paper. This is trying to have a paper that explains that. And it will be published soon. And I would like to acknowledge all of these people who helped me to make this tool. So with that, I would like to thank you for your time. And please feel free to put any questions to the chat.
Awesome. Thank you so much, Mahita. We do have some questions coming through, so I'll give you a minute to take a look as well. And then I will invite Eve and Raphael back to join Mahita for the last Q&A for day 2. So Eve, I think you can take the first question.
Okay, great. So I am kind of tackling 2 at once. We've got a question from Stuart New, which is, do you have any thoughts about using structural models predicted via the in silico approaches like AlphaFold for conformational B-cell epitope prediction? And then we had another anonymous attendee that asked about using AlphaFold predictions for B-cell epitope prediction tools in the near future. So there's 2 parts to this. So there's the idea of general non-antibody specific B-cell epitope prediction, which are the tools that we currently have available on the IEDB. And in that instance, you would use AlphaFold to predict the antigen structure. So those would be the structural models. And as we said during the presentation, I think definitely that's a good idea. Of course, subject to the models looking decent. So definitely have a look at the models and look at the PLDDT that it looks like an acceptable model. And then the second thing, so of using AlphaFold for antibody specific epitope prediction. Yes, that definitely is a viable route for doing B-cell epitope prediction that has been benchmarked by people like Brian Pierce. Obviously, that also takes a lot longer. So in terms of, and there would be license issues for that working within the IEDB. But that is definitely a good line of inquiry for doing antibody-specific B-cell epitope prediction. So I hope that that's answered your questions on that front.
I'll take the next one, Eve. Then I have Emily asking, do we have a mouse TCR-seq results for studying repertoire of different, it changed, different transgenic mice. So does TCRMatch only work for human TCRs, or can we use TCRMatch for mouse data as well? TCRMatch database is only for TCR, for human, but there's nothing, when I showed you on the website, you can switch from IEDB database in CDAR. You can also, in the standalone version of the tool, there's nothing that prevents you from using a murine database that you would have to make, or I would have to check in the IEDB if it's possible to make one for you, but we have to follow this up by email, Emily. And there's a follow-up to your question. You asked if there's a standalone version of the tool. Yes. Can we process TCR data in batch using the standalone? Yes. And yeah, we need to follow up with the fact that it is a murine database, because you also mentioned it's a transgenic rat. So with that, can you use humans? So we need to follow up on this one. Might be a little more complicated.
Awesome. And I've just popped the IEDB help email in the chat, help@iedb.org. So Emily, and then obviously anyone else, if you could contact us there, and then we can use the ticketing system that Bjorn mentioned earlier today. Thank you so much. And Mahita, would you like to select your question?
Yeah, today the question from anonymous say that Bjorn dimension reiterated that there will be another tool with a similar approach to TCRMatch, but for antibody. Contribution information like the data is the train center. So I thought explained about the tool. And the paper contains the information about the training and the OPT submitted by the end of this month. So it will be out in a couple of months. So with that, yeah.
Awesome. Thanks, Mahita. And I know we discussed, I can just repeat some of that. And I'm not sure if this question came through after Mahita's presentation was already partway through, so maybe the person didn't pick up on it. But yeah, Bjorn did mention yesterday that was a similar tool to TCRMatch, but for antibodies. So that was Mahita's presentation. And as she mentioned, a paper is in the works. Hopefully will be completed by the end of the month. So, it will be available to everyone else within the next few months, probably in the new year. So thank you for that. It might have been a timing thing from when that question came through. But Eve, take it away.
Okay, great. So the next question is from Krista McCutcheon, where she says, I find it strange that a single amino acid, the valine in your B cell epitope predicted is in the output. Also, if a reported epitope is really long, e.g. 165 amino acids, does that suggest there's a secondary structure being applied? So yeah, I think I saw your comment, your question, sorry, earlier this morning. So you had your 59 kilodalton protein and you're getting some really long predicted linear epitopes. So the answer to both of these questions, so also the single amino acid and when you have epitopes which are really long, is that there is no, the algorithm just produces for particular sequence windows how likely that is to be an epitope residue. There's no like second step in the algorithm that would filter those predictions to produce plausible like linear epitopes that would be recognized in a completely linear fashion. So of course, if you have 165 amino acid, a long stretch of sequence that wouldn't be plausibly recognized by a single antibody at one time. And so if you are specifically looking for plausible linear epitopes, I would suggest increasing the stringency of the cutoff so that you'll get increasingly small stretches within that 165 amino acids to things that look of a plausible length. So probably between 10 and 20 amino acids. Yeah, so that's how I would interpret the predictions that you have that are clearly not linear epitopes. And yeah, that's me done there.
Okay, so I'll take the next one. Hi, is there any tool to get TCR-seq information of a patient without performing TCR sequences? For instance, I have a bulk RNA info. Can I use this to get TCR-seq? No, you can't use TCRMatch for that. Yeah, maybe out of my league here, saying that maybe Trust4 can do that. And if you do that using Trust4, you can pipe the result of Trust4 into TCRMatch. But TCRMatch, this is out of the scope of what TCRMatch does. Eve, that's you. Are you done?
Oh yeah, is it back to me, or there no?
Yes.
Oh, okay. Sorry, I didn't know if Mahita was going to answer.
Yeah, I got to answer
Sorry,
one
Mahita.
question. There is a question someone at the unknown with between UTPT to compare identical antibodies. The iod classifier could I but like relevant to ever certain about predictions when comparing antibody to itself. We have an explanation right some antibodies when compared to itself with an higher code than others. So that is a good question. Basically, it would depend on it would depend on how similar the antibodies are. So sometimes some antibodies may not be very similar overall in sequence, but they can do and very similar binding specificity. So when we train the models also learn the similarities. So that could be another reason why some person antibody might and lower critical code than others. That answers your question. Yeah.
Okay, and then I'll do another double of very related questions about the training data for Discotope. So the first anonymous attendee asked, for training Discotope, does it matter if the structure was reported in cancer or disease experiments, or did you pool everything you could get? And then the second anonymous attendee said, you mentioned 75 crystal structures of antibody protein complexes to train the model, but how did you test the model? I might have missed this, but did this number change between the two versions of the tool? If so, do you see accuracy improvement? And how do you see the perspective of improving it with more/new structures? Discotope, both Discotope 1 and Discotope 2 were trained on the same set of 75 of the 75 structures. So any performance differences between Discotope 1 and Discotope 2 are actually not the result of an increase in the available training data, but rather modifications to the algorithm itself in terms of stuff like calculating the neighborhood and proximity weighting in the function. And in terms of how and then, as to the first question, whether these are, from cancer or disease experiments, it's sort of agnostic to where these antibodies would be coming from. So it is, as you say, whatever you can, whatever that they could scrape together at that time. In terms of how the model was tested, Discotope 2 was tested on an independent test set of 52 crystal structures where there was no homology in the antigens between the training and the test data. So that was the idea as to how that will generalize in the future. And I'll post a link in the chat to the description on the IEDB of those data sets where you can see the actual PDB IDs. And then, so do you see the accuracy improvement between Discotope 1 and Discotope 2? You do see an accuracy improvement that was reported in the 2012 paper from 0.7 to 0.73. And then finally, the perspective of improving it with more new structures, absolutely. And so Discotope 3 was published this year and was trained on hundreds of structures, but also using a fundamentally different algorithm. And I think I don't know if Bjorn is here, but I can't see him. But in terms of Discotope 3 is something that we will hopefully be able to provide on the IEDB in the future. And there's really significant performance improvements, like ROC AUC of 0.8. So yeah, that's me.
To add on to that, because Bjorn did unfortunately have to drop off, I believe our upcoming next gen release, so after the T cell class 2, we will have a B cell release. And I do believe that the Discotope 3.0 will be included in that one. So yeah.
Great, fantastic. Yeah, so look out for that because that's a really great tool.
That's me. So I have an anonymous attendee asking, the TCR matching tool appears to be comparing amino acid sequences between TCRs and looks like the binding peptides inferring similar TCR sequences bind similar peptides. That is a perfect summary. Like if you switch TCRs by CDR3 betas, that is a perfect explanation of TCR match. But where is the MHC in that equation? Part of the TCR binds certain MHC residues as well, doesn't it? Yes, and it's nowhere. Just like it's TCR match in its current incarnation is not MHC aware. And it also, there's no data about the alpha chain. There's no CDRs 1 and 2. These things are slowly being tested, and they will be incorporated in TCR match if you get better results with them. But in its current incarnation, TCR match is really what you just described. It compares sequences of CDR3 betas and assumes that if they're sufficiently similar, they bind to the same peptide. Mahita.
Hi, the question from 10%, and the question can I use the BCRMatch to find the antibody to find to the same epitope? Yes. What I would like to find out is BCRMatch does not predict the epitope. So you need the antibody with the epitope is known. The BCRMatch. So if our antibody that with the epitope is known, then can compare that antibody with other antibodies whose epitopes are not known. that we can find out that both antibodies have a similar or common epitope.
Okay, great. I'll just answer the next question here. So what does propensity indicate for the B cell epitope? So propensity is used here as a synonym for some measure of how much more likely an amino acid is to be in the epitope versus not in the epitope. So that could be most commonly a log odds ratio, and then you calculate that from some set of structures. So there'll be certain amino acids that have a high propensity, so like polar amino acids, and then certain ones which will have a very low propensity, like cysteine or methionine, are based on their based on the chemistry.
Great. I have one question from GP Shi. Using TCRMatch, if inputting one CDR3 beta sequence, how many epitope sequences may come out? Excellent question. There's no fixed number. It depends on how many epitopes were characterized for that CDR3 beta that is in the IEDB and that TCRMatch finds it sufficiently similar to. So if it's just one epitope that's been characterized for the matching CDR3 beta, then just one. If it's a million, then a million. So, but there is no fixed number. Mahita?
Yeah, would you like to answer another one, Mahita?
There are no consensus.
Okay. So in that case, I'll just answer this question from an anonymous attendee about exporting discontinuous B cell epitopes from the IEDB. So there's no direct link to a PDB or PDB ID, though there are linked 3D structures in the query results. Having to pull the sequence of an epitope and then query PDB and then go back to IEDB for prediction adds a significant amount of time. Is there an easier way to go from discontinuous IEDB entries to either predictor tool? So I think maybe I don't fully understand this question. So please feel free to follow up. So there's some conflation between exporting discontinuous B cell epitopes. You wouldn't need to put those through to the predictor tool. But yes, certainly if there are things that you'd like to see streamlined, please follow up with us.
I have one last question here from Kiran. Since the presence of certain amino acids adjacent to each other can affect protein function and binding affinity, I'm curious about the accuracy of TCRMatch. So specifically, how reliably can TCRMatch identify the most probable epitope that would bind to my TCR sequence if it shares, say, 90% sequence match with a TCR in the IEDB database? Well, in this case, you very likely find a match because you just put a very high sequence similarity. But the thing with the amino acids affecting and shifting by one position, this all happens. And this is why the Kamer approach proved so effective for TCRMatch, because then you compare different Kamers in different positions. And that's why TCRMatch ended up on top as the best method that with the algorithms that we tested.
Great. And then I think, sorry, Nina, do we need to wrap up now?
No, if you want to take that last question, that'd be awesome.
Okay, yeah, just one. So the last question from Shimona Carr, which is based on what is the minimum score cutoff for residues determined for the B cell epitope. So that's something that is determined by the benchmarking results from the original authors, and that there is a reported specificity and sensitivity value for that. Yeah, so there will be a thoughtful default already set for each of these tools. Thank you.
Amazing and perfect timing. Thank you so much, Eve, Raphael, and Mahita for covering all those questions and your presentations. We are out of time, so we will wrap up. Thank you all so much for your time and effort.